Hello and welcome to another very entertaining video. Today's topic, we continue with the topic of metabolism. And on the docket today, we want to understand how we process fats and then kind of continue the story where we left off with sugars as we did glycolysis in the previous video and what comes of the product, which is acetyl-CoA. So if you recall from the last video, we talked about glycolysis. Glycolysis was all about carbs, right? We started with glucose, and through a series of steps, we ended up with pyruvate. And along the way, we generated 2 ATP and 2 NADH. Of course, we made 2 pyruvate in the process. Now, pyruvate, we discussed, had 2 choices, depending on the needs of the cell. Could be converted into lactate, or it could enter the mitochondria, and that's where we're going to follow it. We'll go through the double membrane mitochondria and end up in the matrix. And once inside the matrix, we'll have another redox reaction where CO2 is generated. We also generate per pyruvate more NADH because this is a redox reaction. And what we're left with, pyruvate is a 3-carbon molecule, and now we're left with a 2-carbon molecule, acetate, but it's bound to coenzyme A. So we call this acetyl-CoA. Acetyl-CoA seems to be kind of a hub in metabolism. It regulates a lot of what goes on. Helps us determine whether or not we're processing fats or sugars or protein, whatever. And certainly we can get here, and we'll talk about this in just a second, through the processing of fats. So we talked about carbs, now we're going to talk about fats. This process is called beta oxidation. And we can get here through protein as well. Protein is a little bit trickier. It totally depends on the amino acid we're talking about. We're not going to detail that. But this acetyl-CoA is a central hub and something that we want to focus on. We'll pick this story up at this spot in just a second. What I'd like to do now is turn our attention to this idea of beta oxidation. So how we process fats. And by fats, what do we mean, right? We're talking about triglycerides. And just to remind you about previous units that we've discussed, the triglyceride, of course, is made of a glycerol backbone. Glycerol is a 3-carbon alcohol. And through the hydroxyl groups that were on this backbone, we have formed ester linkages with fatty acids. We'll do the fatty acids in a different color. And that could be any number of carbons, the average 16, 18 carbons in length for us. We'll just kind of abbreviate it. Looks like that. So this is my triglyceride, the glycerol backbone, shown in pink, and 3 fatty acids. And these are both produced and stored primarily in adipocytes, in fat cells in the body. Many of us, of course, are too familiar with storing fat. How do we break it down? Well, there are a couple of different signals that would actually cause the hydrolysis of these triglycerides in the adipose cells. I'm thinking of increased glucagon. This is what we're going to get with low sugar. So if I'm not eating a lot of sugar, then the pancreas releases glucagon primarily to regulate blood sugar, but also to signal, hey, if we're not eating carbs, we've got to switch over to burning fats. We can also see this with epinephrine and norepinephrine. Epinephrine is the adrenaline, right? This is fight or flight response. So if I'm exercising, if I'm running hard, whatever it is that I might be doing, these things can signal the hydrolysis of this ester linkage. We'll draw our scissors here, and this is where we're going to cut, and we're going to liberate these fatty acids. And these free fatty acids are going to be dumped into the blood. The blood's not too happy with something so hydrophobic as a fatty acid floating around, but it gets some help from non-selective carrier proteins like albumin that will help transport these free fatty acids in the blood. And they're going to be picked up primarily by muscle cells if we're thinking about energy. I'm not going to burn fats in neurons in the central nervous system because fats getting past the blood-brain barrier is kind of a hard thing to do. And so while the brain relies on glucose primarily and ketones, if you watch that video, I talk a little bit about that as well. Muscles can very efficiently burn these fatty acids. One of the key differences between fats and sugars is burning fat requires oxygen. This is all oxidative because the mitochondria is essential for the process. And so other cells like red blood cells, for example, that don't have mitochondria, it's just a bag of cells, they can't use fat for energy. So they're limited to glycolysis and sugars. But muscle cells are very good at burning fat because it's such a energy-dense form of energy. So now that we've gotten into the blood, where are we headed? Well, we're going to be picked up by cells, and we're going to be transported into the mitochondria. So let's remind ourselves really quick. The mitochondria is often drawn like this, where we can see two membranes, an inner membrane, an outer membrane. Of course, this central portion is called the matrix. We've got the intermembrane space, this area in here that will be super important as we go on to understand the electron transport chain. Everything that we're going to be talking about today, both beta oxidation and the citric acid cycle, Krebs cycle, that's happening in the matrix. That's happening right in here, right in the middle. And so as these fatty acids come into the cell, again, they're transported inside here. We don't want to detail the specifics of how that transport happens, but they get in there, and this is where we're going to start to break them down. And again, the process that we use to break these down is called beta oxidation. So let's look at a fatty acid. To highlight what we need to see, I need to add the hydrogens, at least to these first few carbons. Because remember, the story that we've been talking about, the story that we've been building is this idea that metabolism is simply a story of carbon oxidation. And as we look at this picture, we recognize that these are carbon-carbon and carbon-hydrogen bonds. These are carbons in their most reduced form. The idea of oxidation is we're going to strip electrons from these carbons, and we're going to use those electrons to run the electron transport chain. And as we look at this, we ask, where are the electrons? Well, they're in the bonds, right? Remember, these covalent bonds are shared electrons. And we've got to figure out how to get those and use those electrons in the electron transport chain. Well, one of the first things that I'm going to do, and this is actually part of getting this fatty acid into the matrix of the mitochondria, I want to bring in a coenzyme A, right? This is the same coenzyme that we saw with acetyl-CoA just a second ago. Coenzyme A is just a carrier. It's a taxi cab. Coenzyme A carries lots of different things, and sometimes you'll see it written like this, CoASH, because the S is a sulfur group that is the functional group by which it can grab on and carry a lot of these things. You're familiar with dehydration synthesis? That's exactly how we're going to attach this. We can see the water right here, H2O, right? So as water leaves, that's going to allow coenzyme A to attach to the carbon double bonded to oxygen through that sulfur group. So now we've got what's called acyl-CoA. It's, again, very similar to acetyl-CoA. Acetyl is a 2-carbon fatty acid, and this is a much longer fatty acid, essentially. That's a decent way to think about it. So we've got our fatty acid bound to coenzyme A, and we're ready to start the process of beta oxidation. One of the first things that I want you to recognize is why we call it beta oxidation in the first place. Well, you're probably familiar with omega fatty acids, right? They're big in the news for health reasons. I need an omega-3 fatty acid or an omega-6 fatty acid. In terms of the Greek alphabet, omega is, of course, at the end. Alpha is at the beginning. Nutritionists, when they talk about fatty acids, they will use omega as a starting point to number carbon-carbon double bonds. So an omega-3 fatty acid is, if I start at the very end carbon and I count backwards, by the time I get to carbon number 3, boom, there's going to be a double bond. Well, we're going to start at the beginning. We're going to start with alpha, and that's going to help us understand what beta oxidation is. This carbon right here, we're not going to count this carbon. This carbon is part of the functional group, and we're going to leave it alone. But this next carbon we call the alpha carbon, which means that carbon number 2 is the beta carbon. Oxidation, again, is the process of stealing electrons off of carbon, and we want to highlight exactly how that's going to happen. So we're going to break that down just into a couple of simple steps. Step 1. This is officially called a dehydrogenase reaction. I don't know that it's important to understand that word too much. simply what are we going to do? We are going to form a carbon-carbon double bond. But because we're super interested in the electrons, it's important to go through and watch all of the bonds that we're playing around with. So let's start by using these electrons right here that are attaching this carbon to the hydrogen, and let's move them. Let's move them right up there to form this carbon-carbon double bond. There's a couple of things that I hope you can see really fast. Number one, this is going to leave behind an H+. We stole the one electron that hydrogen brought, and we used that in that carbon-carbon double bond. So now we have this extra H+, and we'll talk about what to do with that in just a second. The next thing I want you to see is that our beta carbon is now making 5 bonds. And of course, that can't work, right? Carbon makes 4 bonds. And so we are going to remove this hydrogen, but recognize what comes with the hydrogen. This hydrogen is accompanied by the 2 electrons making up that covalent bond that was holding it to the carbon. So here's hydrogen. We're going to draw it this way, where he's got 2 electrons and a negative charge. And we call this hydride ion. It's another ion of hydrogen. So let's move hydride ion down here. We've actually made a trans fat, which is super interesting. And there it is, because of this carbon-carbon double bond between the alpha carbon and the beta carbon of this fatty acid. Remember, we're trying to steal electrons. Well, guess what? We have a lot of electrons right there, right? Those 2 extra electrons that we kind of hold off. How do we store them? What are our electron carriers? What are our taxi cabs for electrons? Well, in this case, FAD is going to come by and pick up both of those, and we end up with FADH2. And where's FADH2 headed? Over to the electron transport chain, because now I can use those electrons to run that system that's going to help me make ATP. So all of that came because of this first step where we had this carbon-carbon double bond. All right, step 2. Think about ramen noodles for a second, right? One of the easiest things in the world to make. And what did the instructions say? Just add water. What am I going to do with water? Well, I'm going to use water to go in and break that carbon-carbon double bond that I just made. Remember, if we think about what water looks like, we've got the oxygen, 2 different hydrogens, 2 polar covalent bonds. Polar covalent bonds are strong bonds, right? That oxygen really has a pull on those electrons. We can use this in the oxidation process. It's an energy favorable reaction for carbon to bond to oxygen rather than to hydrogen, which is how we can steal the energy out of it. To do that, we are going to kind of have to split water. So let's think about water in these 2 different ways. And first, let's take this hydrogen with its electrons and let's add them to the alpha carbon. So there's the 2 electrons. There's the hydrogen added to the alpha carbon. Of course, now that double bond between those 2 carbons doesn't work because we're making 5 bonds. And so we need to move those electrons. Let's swing them up here. And we can now add the rest of the water molecule, the O and the H, to the beta carbon. I hope again you can see this is beta oxidation. I'm oxidizing that beta carbon. It was a carbon-hydrogen bond, and now it's a carbon-oxygen bond. A more energy-favorable bond, and with the energy that was released in making that switch, what have we done? We've stolen those electrons, and they're loaded onto that FADH2 that we had carry off the hydride ion in step 1. All right, that brings us to our 3rd step, and we'll call it break with sulfur. A more technical term would be thiolysis, but whatever. We recognize, talking earlier about coenzyme A, the functional group on coenzyme A is sulfur. So let's move this fatty acid over, and we're going to bring in another coenzyme A. And with that sulfur, because it's a more energy favorable bond, we're going to bond to the beta carbon. Imagine that line being a new covalent bond. Well, a couple of things are going to happen. First of all, that's going to displace both this bond and this hydrogen. So out comes the hydrogen, this hydrogen ion. We're going to take that bond and we're going to swing it to the other side of carbon, and we're going to use it to form a double bond with oxygen. We can do that because we actually end up cutting this bond right here and separating this carbon chain. So now that we're separated, we need to complete the octet rule for carbon. We do have this extra hydrogen over here, right? So let's use this hydrogen to fill in right here. And what have we just made? This molecule right here is acetyl-CoA. The hub, the center portion of this metabolism that we've been talking about and how we introduced this video. We've now made an acetyl-CoA. And we're going to use that acetyl-CoA in the Krebs cycle. So off it goes to the Krebs cycle. Now look at what we have left. Let's clean this up a little bit. Now we've got the coenzyme A attached to a new carbon. We've removed acetyl-CoA. There's one more thing about this picture that I hope you notice. Right here on top, how many bonds can oxygen make? Oxygen can only make 2 bonds. And both of those are now to the carbon. Of course, hydrogen feels out of place. So what happens? The hydrogen leaves. But not just the hydrogen. What does it carry with it? It carries those 2 electrons, those 2 right there, that are making up the covalent bond that held it to oxygen. So what left? Hydride ion. So let's move hydride ion out here. Of course, these are very high energy electrons. And we have another carrier in the form of NAD+. That's going to come pick up this H and its 2 electrons, and we get NADH. So when we break with sulfur, what do we end up with? This is another redox reaction, and we end up with 1 NADH. Lastly, there's one more thing that I want you to recognize. If we look at this molecule that we're left with, I hope you recognize that it is an exact duplicate of what we started with, except we're 2 carbons shorter. This becomes my new alpha carbon. This becomes my new beta carbon. And I do it again. I repeat the process of beta oxidation, and I'll cut there and I'll make another acetyl-CoA. And then I'll cut there and make another acetyl-CoA. And then I'll cut there and make another acetyl-CoA. And then on this last one, I'll cut there and I will make 2 acetyl-CoA. That's what's left over. So in summary, when we think of beta oxidation, per reaction, what are we generating? We're generating 1 FADH2, 1 NADH, and 1 acetyl-CoA. Except at the very end, when we take that 4-carbon structure and cut it in half, now I have 2 acetyl-CoA. And the acetyl-CoA, of course, are going to go to where we're going now, which is the Krebs cycle, for further oxidation to get all the extra energy out of that. All right, here's a picture of the Krebs cycle, showing all of its intermediates. Certainly at this level, we don't need to know the names. We certainly don't need to be familiar with the structures, but I bring the structures in for a purpose that I'll show you in just a second. First, I want to direct your attention to the left side. This is the beginning process, right? All of that acetyl-CoA that we've been generating, it's right there. So this molecule shown in blue is acetyl-CoA. Coenzyme A through the sulfur group attached to 2 carbons. And the very first step in the process is attaching that 2-carbon acetyl group onto this 4-carbon molecule called oxaloacetate. And with 2 carbons there and 4 carbons there, what do we end up with? We end up with citrate, which is a 6-carbon molecule. Now, some of the things that are interesting to look at. Remember, we've still got energy stored in those carbon-carbon bonds. There's still redox reactions to be had. We can see that here, right? Where we go from a 6-carbon molecule, isocitrate, to a 5-carbon molecule, alpha-ketoglutarate. What do we generate in the process? We generate an NADH. And remember, that's where my electrons are stored, and I'm going to use that to run the electron transport chain. We also generate, as a waste product, carbon dioxide. That carbon in carbon dioxide represents carbon in its most oxidized form. Metabolism is a story of carbon oxidation. And when there's no more energy left in those carbons, we breathe it out as carbon dioxide. We see that same thing here where we generate another NADH, and sure enough, there's another carbon dioxide generated. And now we're down to a 4-carbon molecule. There's still some redox that we can do, and this is where I think it's really interesting. If you recall the steps that we went through for beta oxidation, I think it's fascinating to see that we're going to repeat the exact same things. Specifically, going from succinate to fumarate, from here to here, what do you see? You see the presence of a carbon-carbon double bond. What was the first step in beta oxidation? It was the formation of a carbon-carbon double bond. That kicked off H+ and a hydride ion, and what picked it up? FADH2 was made as FAD picked up those 2 hydrogens and the electrons they were carrying. Step 2 of beta oxidation, just add water. Oh, look at that. There's water coming in. The carbon-carbon double bond is lost, and we can see water on this molecule. Right? There's the OH portion of it right there. The rest of water would be up here, the hydrogen that's now bound to this carbon. Now, on this last one, we're not going to actually split with sulfur like we did in beta oxidation, but it is the same process where we're going to take that hydroxyl group, that OH, and we're actually going to convert it into a double bonded oxygen. And in the process, we kick off hydride ion, and where do we pick it up? Right there as NADH. So that's why I wanted to show you these structures, because I think it's fascinating that the same process that we saw with the breakdown of fats is the same repetitive, evolutionarily conserved process that we're using on the back half of the Krebs cycle. It's conserved, it's repeated. And in the end, what do we get out of it? Per cycle, in other words, per one revolution, we generate 3 NADH. These are high energy electrons that electron carrier is holding onto, and this is what we're going to use to run the electron transport chain, which we'll talk about in the next video. We can see them here, 1, 2, and 3. We also generate 1 FADH2. Same principle as NADH, only not quite as cool. This is like an Uber driver, right? But there it is. There's the FH2 that was generated. Because this is all about energy, we have to recognize, too, that in the process we generated 1 ATP. This shows it right here, where we actually generated GTP, and then we can transfer it onto ADP to make ATP. Now, it doesn't matter that we understand the specifics. Just recognize that in this step, we actually do generate an ATP. And I think most importantly, even though it's a waste product, we generate 2 carbon dioxide. And we saw those here and here. So why is that important? Because remember, this acetyl group, the acetyl-CoA that came in, 2 carbons right there. And while those aren't the exact same carbons that were oxidized in this cycle, the net result is the same. 2 come in, 2 are converted to carbon dioxide, right? This is the ultimate carbon oxidation. And we recycle back to oxaloacetate to pick up the story. Oxaloacetate's going to bind another acetyl-CoA, pick up those 2 carbons, and start the process again. So every time you exhale, right, take a deep breath and exhale, all of that carbon dioxide that came out of your mouth, this is the primary source of it. The other step where we talked about pyruvate oxidation, we also showed that there was a carbon dioxide emitted there. That's where we make CO2. And every time we breathe out, it's because of the CO2 that's produced in these steps. Hopefully that gives you a good sense of what Krebs cycle is all about. It's similarity to beta oxidation of fats. Both are happening in the matrix of the mitochondria. Both heavily involve redox reactions. Both generate NADH and FADH2. And that's so important because these 2 carriers right here are loaded with electrons. And I am going to use those electrons. Remember, NAD and FAD, these are just taxi cabs. They pick up and then they drop off. We've picked up all these electrons, and now we're ready to drop them off at the electron transport chain where we're going to take all of this energy and really start to make a lot of ATP.